Hello students, welcome to the lecture on biotechnology and its applications. And after this lecture, we will be able to learn the following objectives. Describe the recombinant DNA technology, discuss about agriculture and industry, explain about genetically modified GM organisms, describe about insulin and BT cotton. Let's start with a brief introduction to biotechnology. Biotechnology is technology based on biology, especially when used in agriculture, food science, and medicine. The UN Convention on Biological Diversity has come up with one of many definitions of biotechnology. Biotechnology means any technological application that uses biological systems, living organisms, or derivatives thereof to make or modify products or processes for specific use. Biotechnology is also commonly associated with landmark breakthroughs in new medical therapies to treat diabetes, hepatitis B, hepatitis C, cancers, arthritis, hemophilia, bone fractures, multiple sclerosis, cardiovascular as well as molecular diagnostic devices that can be used to define the patient population. Perception is the first drug approved for use with a matching diagnostic test and is used to treat breast cancer in women whose cancer cells express the protein HER2. What then is biotechnology? The term brings to mind many different things. Some think of developing new types of animals. Others dream of almost unlimited sources of human therapeutic drugs. Still others envision the possibility of growing crops that are more nutritious and naturally pest resistant to feed a rapidly growing world population. This question elicits almost as many first thought responses as there are people to whom the question can be posed. In its purest form, the term biotechnology refers to the use of living organisms or their products to modify human health and the human environment. Recombinant DNA technology is a technology that allows DNA to be produced via artificial means. Basically, these techniques are used to achieve the following. Study the arrangement, expression, and regulation of genes. Modification of genes to obtain a changed protein product. Modification of gene expression either to enhance or suppress a particular product. Making multiple copies of a nucleic acid segment artificially. Introduction of genes from organism to another, thus creating a transgenic organism. Creation of organism with desirable or altered characteristics. The video here will show us the process of DNA recombination.
Recombinant DNA technology is a process used in the field of biotechnology to genetically manipulate cells to carry out a function or produce a product. First, the gene of interest is isolated from the original cell and amplified in a process known as PCR. Each round of PCR doubles the amount of the gene present. Usually 30 cycles of PCR are performed, amplifying the gene by a factor of 2 to the 30th power. Next, the gene is joined to a plasmid vector which contains genes to allow for the selection of successful recombinants, usually an antibiotic resistance gene. This step is known as ligation and is done due to the activity of DNA ligase. In TA cloning, the vector is designed to take advantage of the fact that the Taq polymerase enzyme used in PCR adds an extra A to the gene product on the 5' end. It does this by having a T residue overhangs on the 3' ends, which act as sticky ends. Before carrying out this procedure, the most suitable cell type is selected, which depends on the gene and the vector being used. This cell type may be treated further to make it suitable, i.e. rendered competent. For example, the addition of calcium ions and a heat shock. The vector can be inserted into the cell by three different mechanisms. Electroporation, which is where a voltage is applied to the culture, causing pores to form in the cell membrane. Infection, where the vector is encapsulated in a virus particle which infects a cell. We refer to this as infection. Or, if using bacteria, we can induce them to take up the plasmid by a heat shock, for example. This is referred to as transfection. The transformed cells are then cultured, and then successful transformed cells are picked out due to the expression or non-expression of certain genes that are encoded on the plasmid. For example, blue-white selection. Tests can be performed to see if the cell contains and expresses the gene of interest. For example, phenotype testing, restriction enzyme analysis, and or sequencing. There you go. Now your cell is ready to produce or perform what you designed it to do.
Let us take a look at the three options that can be thought for increasing food production. Agrochemical based agriculture, organic agriculture, and genetically engineered crop based agriculture. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Increased yields have partly been due to the use of improved crop varieties, but mainly due to the use of better management practices and use of agrochemicals. Plants, bacteria, fungi and animals whose genes have been altered by manipulation are called genetically modified organisms, GMO. GM plants have been useful in many ways. Genetic modification has made crops more tolerant to biotic stresses, cold, drought, salt, heat, reduced reliance on chemical pesticides, pest-resistant crops, helped to reduce post-harvest losses, increased efficiency of mineral usage by plants. This prevents early exhaustion of fertility of soil, enhanced nutritional value of food, example, vitamin A enriched rice, pest-resistant plants. Several nematodes parasitize a wide variety of plants and animals, including human beings. A nematode, Meloidigene incognita, infects the roots of tobacco plants and causes a great reduction in yield. A novel strategy was adopted to prevent this infestation, which was based on the process of RNA interference, RNAi. RNAi takes place in all eukaryotic organisms as a method of cellular defense. This method involves silencing of a specific mRNA due to a complementary dsRNA molecule that binds to and prevents translation of the mRNA silencing. The source of this complementary RNA could be from an infection by viruses having RNA genomes or mobile genetic elements, transposers that replicate via an RNA intermediate. Using agrobacterium vectors, nematode-specific genes were introduced into the host plant. The introduction of DNA was such that it produced both sense and antisense RNA in the host cells. The relationship between agriculture and industry is very important in any economic development. The rise in the demand for food and in agricultural prices have stimulated the development of agriculture, its capacity to increase its purchases from industry, and its deliveries of products to the market. Increased consumption of producer goods has been sustained by increased agricultural prices and state subsidies for the purchase of equipment more than by advances in agricultural productivity. The growth in agricultural production has been obtained by extending crops rather than by increased yields. For thousands of years, farmers and plant breeders have been changing crop plants to improve characteristics such as size, resistance to disease, and taste. Plants which grow well, have a higher yield, or taste better are selected and bred from. This is still the most widely used technique for developing new varieties of a crop and is limited by natural barriers which stop different species of organisms from breeding with each other. Genetic modification is very different to these traditional plant breeding techniques. It is a technology which allows scientists to take genes from one organism and put them into another. Transferring DNA and genes from one organism to another is a difficult and fairly haphazard procedure. Scientists have voiced concern that such disruptions could lead to unexpected toxins being produced or to changes in the levels of nutrients and naturally occurring toxins. There are examples of genetic modification changing plants in entirely unexpected ways. For example, when researchers in Germany tried to boost the starch content of potatoes using genes from yeast and bacteria, they found that the starch content actually fell and other unexpected compounds were produced. There are three types of tendencies to provoke allergic reaction: allergenicity, gene transfer, and outcrossing. Allergenicity. As a matter of principle, the transfer of genes from commonly allergenic foods is discouraged unless it can be demonstrated that the protein product of the transferred gene is not allergenic. While traditionally developed foods are not generally tested for allergenicity, protocols for tests for GM foods have been evaluated by the Food and Agriculture Organization of the United Nations, FAO, and WHO. Gene transfer. Gene transfer from GM foods to cells of the body or to bacteria in the gastrointestinal tract would cause concern if the transferred genetic materials adversely affects human health. This would be particularly relevant if antibiotic resistance genes used in creating GMOs were to be transferred. Although the probability of transfer is low, the use of technology without antibiotic resistance genes has been encouraged by a recent FAO-WHO expert panel. Outcrossing. The movement of genes from GM plants into conventional crops or related species in the wild, referred to as outcrossing, as well as the mixing of crops derived from conventional seeds with those grown using GM crops, may have an indirect effect on food safety and food security. This risk is real, as was shown when traces of a maize type, which was only approved for feed use, appeared in maize products for human consumption in the United States of America. Several countries have adopted strategies to reduce mixing, including a clear separation of the fields within which GM crops and conventional crops are grown. The government will only agree to the planting of GM crops, the release of other types of GM organism, or the marketing of GM food or feed products if a robust risk assessment indicates that it is safe for people and the environment. GM product applications should be assessed for safety on a case-by-case basis, taking full account of the scientific evidence. The government supports farmers having access to developments in new technology and being able to choose whether or not to adopt them. Many different types of GM crops are now being developed. In the UK, all the GM crops that are close to gaining commercial approval to be grown by farmers are herbicide, weed killer tolerant. These have been engineered to be tolerant to powerful herbicides which kill all plants. This means that only the crop can survive being sprayed and all other plants in the field die. Genetic engineering is imprecise and unpredictable. By inserting genes from organisms which have never been eaten as food, new proteins are introduced into the human and animal food chains. There is concern that these could cause allergic reactions or other health effects. The safety of GM foods depends on government assessment of tests conducted by the GM companies themselves. Most of these tests have never been published or subjected to independent peer review. It is often claimed that one reason for having GM crops is that they will help to feed the world's growing population in the coming century by increasing yields and fighting crop genetically modified crops and food diseases. However, many people in the world are suffering from malnutrition and hunger because they cannot afford to buy food, not because it is unavailable. Complex social, political, and economic forces affect how people have access to land, money, and resources. Several countries that have faced serious food shortages have raised concerns about the use of GM crops in food aid. These concerns have focused on the health and environmental effects of introducing GM crops and food during emergency situations, often in countries lacking biosafety regulations. One area of concern is the unknown impact of feeding GM food as the main constituent in the diets of immune-compromised populations through hunger and often HIV/AIDS. Scientists have already raised similar concerns about the feeding of GM in high quantities to both farm animals and babies. Scientists working for the Zambian government, which rejected GM food aid, stated that while it is often said that GM maize is consumed by millions of Americans, it was noted that it is eaten in highly processed form and is not a staple food in the USA. In Zambia, maize is the staple food and is usually the only carbohydrate source. Biosafety is a concept that refers to measures put in place to mitigate or protect human health and the environment from possible adverse effects of the products of modern biotechnology. The Cartagena Protocol on Biosafety provides a comprehensive and holistic regime designed to ensure that the development, handling, transport, and use of products of modern biotechnology are undertaken in a manner that maximizes benefits while preventing or reducing risks to the environment and human health. The protocol is a subsidiary agreement to the UN Convention on Biological Diversity (CBD). Kenya signed the Biosafety Protocol in 2000 and fulfilled the ratification requirements in 2003. One of the key obligations expected from the parties to the protocol is promotion and facilitation of public awareness, education, and participation in biosafety activities as stipulated in Article 23. The National Council for Science and Technology (NCST), established under the Science and Technology Act Cap 250, Laws of Kenya under the Ministry of Higher Education, Science and Technology, is the designated authority that coordinates all matters pertaining to biosafety in Kenya. The National Biosafety Committee (NBC) is the technical arm of the Council charged with the mandates of overseeing coordination and implementation of biosafety issues. BT cotton is a genetically engineered form of natural cotton. The main advantages of utilizing biotechnology in agriculture are the possibilities of increase in productivity through the use of newer varieties that possess properties such as resistance to pests, diseases, and other stressful conditions like drought, salinity, or waterlogging. The toxin in Bt cotton exists in nature within the microorganism Bacillus thuringiensis. The Japanese bacteriologist Ishiwata Shigetain first discovered it in 1901. Subsequently, in 1915, a German scientist named Ernst Berliner isolated this toxin from a dead moth in Thuringen region of Germany, and thus the name Bacillus thuringiensis.
Impacts of Bt cotton: Gossypium hirsutum, Gossypium barbadense, Gossypium arboreum, Gossypium herbaceum. These are common cotton varieties. Differences between Bt and non-Bt cotton. Bt cotton during 2010 in India on the superiority of Bt cotton over conventional cotton. The plant protection chemicals had significant positive influence in Bt cotton production, while it was negative, non-significant in non-Bt cotton farms. The decomposition analysis per acre output of cotton in Bt farms was higher by 16% over non-Bt farms. Importance of Bt cotton. Cotton has been genetically modified to resistant bollworms. Cotton that has been engineered to contain a gene from the bacterium Bacillus thuringiensis, making it resistant to attacks by bollworm. Bt cotton has decreased pupation rate compared with non-Bt cotton. And it is to reduce the heavy reliance on pesticides. Adoption of GM cotton. This is a picture of GM cotton in USA by 2002 to 2012. GM crops raise farm incomes. Global cotton adoption of 80% increased compared with the 64% planted to Bt cotton in 2010. In 2011 Bt cotton level. The area planted to be Bt cotton globally in 2011 was record 24.7 million hectare, up by 3.7 million hectare. At 24.7 million hectare, Bt cotton planting adoption rate is 82% increase in 2009. In 2011, Bt hybrid cotton in India, the largest cotton growing country in the world. Bt cotton increasing by an impressive 13% gain between 2010 and 2011, which is reached 86% in 2010. In 2011, 13 countries growing in Bt cotton. Total of 13 countries growing in Bt cotton in 2011. Biotech cotton traits. Insect resistant traits. The third most dominant biotech crop was Bt cotton, which is equivalent to 11% of the global Bt area since 2010. Bt cotton was planted in 11 countries. Herbicide tolerant traits. The eighth most dominant trait was Bt cotton, occupying 1.8 million hectare or 1% of all Bt crops by globally. In herbicide tolerant cotton was planted in 7 countries.
Bt maize is another Bt crop on which research is being conducted. It was grown on 7.3 million hectares worldwide in 2000. It makes up 20% of the total transgenic crop grown in the world. The net gain in producing this crop from 1997 to 1999 was $20 million. Transgenic crops are grown in many countries like Australia, South Africa, Mexico, Spain, Portugal, Romania, Ukraine, USA, China, etc. In China, research on Bt cotton first began in 1991 and subsequently commercial growing of this cotton started in 1998. The Chinese situation is very similar to that of the Indian situation because both countries have lots of small farmers and scattered land holdings. In the United States, research on Bt cotton began in 1989 and commercial growth began in 1995. Bt cotton has proved to be very beneficial to the US farmers as well. An estimated 260 million pounds extra cotton was produced in 1999 and revenue increased by approximately $99 million. The average insecticide application also decreased from 10 units through 2 units to 0 units. Management of adult onset diabetes is possible by taking insulin at regular time intervals. Insulin used for diabetes was earlier extracted from pancreas of slaughtered cattle and pigs. Insulin from an animal source, though, caused some patients to develop allergy or other types of reactions to the foreign protein.
The date was the 14th of October 1980 in the New York Stock Exchange. The launch of a small biotechnology company called Genentech sparked frantic business. Its share price rocketed from $35 to $88 within the first 20 minutes of trading. At the end of the day, each Genentech share was worth $71.25. Why was there so much interest in a 4-year-old Californian company specializing in genetic engineering? 2 years previously, scientists at Genentech had isolated the human genes which carry the code that enables particular cells of the pancreas to produce the hormone insulin. They had learned how to transfer the genes from these cells into bacterial cells. The bacterial cells had been genetically engineered to contain human insulin genes. The genes continued to control the production of insulin, even though they had been moved from one type of cell, a pancreatic cell, to another type of cell, a bacterial cell. When bacterial cells multiply, the genes which have been transferred into them also multiply. Bacterial cells multiply very rapidly, and large numbers of cells soon build up. In the case of bacterial cells genetically engineered to contain the human insulin genes, this means large amounts of insulin can be made very quickly. The conditions inside huge fermenters encourage the rapid multiplication of bacteria. The fermenters are filled with a solution of all the substances the bacterial cells need to grow. In the case of bacterial cells genetically engineered to produce insulin genes, the insulin the cell makes is released into the solution. It is separated from the solution, purified and packaged, ready for use. Why should we want to transfer insulin genes from the cells of the human pancreas to bacterial cells? Insulin controls the level of sugar in our blood. Some people do not produce enough insulin and suffer from diabetes. Daily injections of insulin control the condition. Before genetic engineering, people suffering from diabetes depended on insulin sourced from cows and pigs. This was unacceptable to vegetarians and people with certain religious beliefs. It was expensive and sometimes caused allergies. All of these problems were overcome when genetically engineered insulin became available to treat diabetes. It is cheap and safe to use because chemically it is the same as insulin our cells produce naturally.
Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child embryo. Here genes are inserted into a person's cells and tissues to treat a disease. Correction of a genetic defect involves delivery of a normal gene into the individual or embryo to take over the function of and compensate for the non-functional gene.
A little bit invasive. In gene therapy, doctors are basically hacking the DNA of a living human. Using genetically engineered retroviruses called vectors, scientists infect human cells. The retrovirus can be programmed to carry a gene or a little bit of DNA that will overwrite the messed up mutation and make it work properly. It was first tried on a young girl in 1990, and despite some early failures, it has the potential to revolutionize treatment of genetic disorders. The journal Science describes one of the recent successes that gene therapists say was really exciting. A few children were born with metachromatic leukodystrophy, which causes a defective immune system and some brain disorders, and kids who have it usually don't live past the age of 5. Bone marrow contains stem cells. The cells normally produce red blood cells, but they can be reprogrammed using gene therapy. It's a little risky, but it can work. Taking bone marrow from these kids, doctors were able to infect the cells with a retrovirus and replace the stem cell's mutated gene with a repaired gene. Then they re-injected that back into the patient, and the fixed cells multiplied, and as of the time we filmed this, the patients are all in good condition and are heading to kindergarten at the time that others with that disease can't even speak. There may be future side effects, but they seem pretty happy with the result at the moment. I mean, I would be. Science just helped some kids. Woo! It's not just useful in children. Scientists have also used gene therapy on dogs to cure them of type 1 diabetes, with 2 of their doggy patients still alive years later. The treatment involved injecting 2 things into dogs' muscles, one gene to sense glucose and an enzyme to dictate glucose absorption. Scientists don't have to target our DNA. They can also use gene therapy to target the DNA of cancer cells. It's like they gave cancer cancer. You've seen this before if you've been following D News. A protein called CD47 is like a passport that tells your immune system not to attack a cell. Normally, cancer produces a ton of CD47. Using gene therapy on the cancer, scientists turned off that cell production and let the immune system blow it out of the sky like a decoy.
Now, in the end, let us summarize what we have learnt in this lecture. In its purest form, the term biotechnology refers to the use of living organisms or their products to modify human health and the human environment. Recombinant DNA technology is a technology that allows DNA to be produced via artificial means. There are 2 main types of cloning that recombinant DNA technology is used for: therapeutic cloning and reproductive cloning. The Green Revolution succeeded in tripling the food supply, but yet it was not enough to feed the growing human population. Agrobacterium is used as natural genetic engineer because of its ability to deliver gene of interest to host genome. RNA interference involves silencing of a specific mRNA due to a complementary dsRNA molecule that binds to and prevents translation of the mRNA. Biosafety is a concept that refers to measures put in place to mitigate or protect human health and the environment from possible adverse effects of the products of modern biotechnology. Bt cotton is a genetically engineered form of natural cotton. Gene therapy is a collection of methods that allows correction of a gene defect that has been diagnosed in a child embryo.